90%. I mean, just think about that number for a second. If you spent, 10 years and billions of dollars building literally anything else, and it failed 90% of the time, you would go bankrupt overnight. But in the pharmaceutical industry, that staggering failure rate in clinical trials hasn't really been a scandal. It's just been the accepted cost of doing business. You grab a chemical, you hope it works, and like 9 times out of 10, it completely fails.
Right, yeah. And the historical baseline for drug development has always been this incredibly stochastic trial and error process. I mean, the human brain, even when you augment it with traditional supercomputers, it simply cannot integrate the sheer volume of multi-omics data that you need to predict these really complex biomolecular interactions. Not with absolute certainty, anyway. We have basically just been playing a numbers game.
And today, we are taking a deep dive into a stack of architectural analyses and benchmarking reports that suggest that numbers game is, well, it's finally over. Our sources today are detailing this massive transition into an era of what they call scientific intelligence. Specifically, we're looking at something called the Pharmaceutical Superintelligence Framework, or PSI, and we're also looking at a gap analysis that benchmarks this exact AI suite against 22 global grand challenges in drug discovery. So the mission of this deep dive is really to figure out if we are actually entering a prompt-to-drug reality, or if autonomous AI cures are still just science fiction.
Yeah, and to really understand the magnitude of the shift, we have to look at why that historical 90% failure rate happens in the first place, right? The sources point to 2 fundamental bottlenecks. First is biological target identification, which essentially means we just aim the drug at the totally wrong part of the disease. And the second is suboptimal lead optimization, meaning, we actually found the right target, but the drug we built to hit it was fundamentally flawed.
Right, and this impacts you listening right now. I mean, you may not be a biochemist, obviously, but a system that can compress a 10-year trial and error nightmare into a hyper-targeted, months-long process, that fundamentally alters the future of your healthcare. We used to search for a drug like someone trying to find a specific grain of sand on a massive beach while completely blindfolded. The PSI suite is essentially like taking off the blindfold and giving you a metal detector, a GPS, and an entire excavation team.
That's a great analogy. The capability leap is just profound. But, it does introduce a massive computational frontier. AI has become, incredibly proficient at predicting the static structures of molecules. The real challenge and kind of the central tension of our sources today is predicting how those newly generated structures behave dynamically inside a living, breathing human body.
Okay, so before we get into the gaps where the AI struggles with that dynamic stuff, we really need to establish how this PSI framework actually operates. Because the sources describe it as a system of systems. It's not just, one massive chatbot where you type, "please cure cancer," and it spits out a pill. It's highly stratified.
Exactly. It operates much more like a massive corporate hierarchy.
Yeah.
So at the very top, you have the user level. You can think of this as the CEO of the operation. A human researcher inputs a natural language objective, for instance. Find a compound that inhibits this specific protein in this specific type of cancer. The user level then translates that broad human goal into a really high-level technical research plan.
And then it just delegates everything down the chain.
Precisely. It passes the plan down to the cohort level, which is basically middle management. These are highly specialized groups of AI agents strictly focused on distinct scientific disciplines. So you have a biology cohort, a chemistry cohort, a process engineering cohort. They take the CEO's broad plan and break it into specialized, actionable tasks. And then finally, at the base, you have the agent level.
The factory floor workers.
Right, the operative primitives. They execute the discrete technical tasks like running a multi-day molecular dynamic simulation, or even actively controlling physical robotic arms in a wet lab through API integrations.
Okay, wait, I'm trying to wrap my head around this. If you have all these different AI agents specialized in totally different disciplines, they're essentially speaking different scientific languages, right? I mean,
Right.
A biology model looks at genomics, but a chemistry model is looking at atomic bonds.
Yeah.
How do they not just create a fragmented, walled garden of messy data? Like, if a simulation at the bottom fails, how does the top even know what went wrong?
That's a massive hurdle, yeah. But the orchestration layer relies on these standardized communication protocols. The sources specifically reference frameworks like Google's A2A or Anthropic's MCP. What's fascinating here is that these protocols force every single agent, regardless of its underlying architecture, to translate its findings into a universal, predictable messaging format.
Oh, wow. So they basically force them all to speak a common lingua franca.
Exactly. And because of this shared context, an insight from, say, a late-stage manufacturing simulation run by the engineering cohort can instantly flow backward to the chemistry cohort, allowing them to adjust the molecular design in real time.
So the whole organization literally learns at once. That's wild. And that interconnectedness basically starts with the biology cohort, trying to figure out what to even aim at in the first place. The sources highlight a system here called TID Pro, or Target Identification Pro. And the numbers here are crazy. It boasts a 71.6% clinical target retrieval rate. To put that in perspective for you, frontier large language models generally score somewhere between 15 and 40% on the exact same task.
Yeah.
But the report says TID Pro crushes them by using a positive unlabeled learning framework. What does that actually mean in practice?
So it addresses a massive data problem in biology. We only have a few definitively proven, highly successful drug targets. Those are the positives. But the vast majority of proteins and genes in the human body are unlabeled. We simply don't know if targeting them will cure a disease or just do absolutely nothing. Standard AI really struggles when it doesn't have clear negative examples to learn from.
Right, because it doesn't know what a bad target looks like.
Exactly. Positive unlabeled learning is like an AI trying to build a highly effective spam filter when it only knows what, say, 5 confirmed spam emails look like, and the rest of the inbox is just this massive pile of unknowns. It has to deduce the hidden structural patterns of a successful target from a total sea of ambiguity.
And it does this by cross-referencing, right? Because the breakdown shows it analyzes 22 distinct feature scores. It takes 12 ONIC-based metrics, so data from genomics and proteomics, and then it marries them with 10 text-driven metrics from decades of scientific literature.
Yeah, and the context dependency is what gives it that massive 71% success rate. TID Pro recognizes that the rules change depending on the disease it's looking at. If it's looking for an oncology target, it weighs mathematical matrix factorization of protein data really heavily. But if it is researching a neurological condition, it shifts its attention to literature density scores, prioritizing what decades of human research texts suggest might be relevant.
It's literally cross-referencing the DNA evidence from a crime scene with witness testimonies from the library. But finding a statistical correlation, saying this protein is usually present when this disease is present, that isn't enough to justify a billion-dollar drug synthesis program. Correlation doesn't equal causation.
Which necessitates the next agent in the sequence, BioReason Pro. This agent provides the actual mechanistic justification. It deduces what the protein is doing on a molecular level. There's this fascinating case study in the architectural analysis regarding a protein called CFA61. Standard AI models were completely misled by it.
Because it looks exactly like an enzyme, right? The notes mentioned it has a catalytic Rossmann-like fold.
Right. A Rossmann fold is a very specific structural shape that almost always indicates a protein is an enzyme, meaning it actively sparks chemical reactions. Earlier models saw that shape and just assumed the drug needed to stop a chemical reaction. But BioReason Pro looked deeper at the structural reasoning and completely overrode that assumption. It correctly identified that despite the shape, CFAP61 is actually a non-enzymatic scaffolding protein. Its job is purely structural support, not chemistry.
Wait, really? So if human researchers had just listened to the earlier AI models, they would have spent years and millions of dollars designing a drug to stop a chemical reaction that wasn't even happening in the first place.
They would have been entirely blind to the actual mechanism of the disease. Once BioReason Pro establishes the true function, it uses internal attention mechanisms to map the protein surface and localize the exact contact residues. It outputs what the system calls hotspot tokens. Basically, it tells the downstream AI exactly which microscopic groove on the protein surface to target.
Okay, so BioReason Pro has pinpointed the exact hotspot. We know where the bullseye is, but knowing where the bullseye is doesn't mean you actually have the dart to hit it. How does the chemistry cohort go about building the actual molecule?
Well, this relies heavily on the Isomorphic Lab's drug design engine, or IsoDDE. The benchmarking data shows it is 1.5 times better than established methods at finding ligand-binding pockets. But its real breakthrough, its crowning achievement, is predicting cryptic pockets.
Oh, let's linger on cryptic pockets for a second, because the physics here are just wild. Proteins aren't static statues, they wiggle and move. A cryptic pocket is essentially like a hidden door in a room that only appears when you lean your entire body weight against one specific wall.
That's spot on. In structural biology terms, when the protein is just floating alone, it is in its apostate. The binding site is completely absent, the door is hidden. It only forms when a drug, a ligand, physically interacts with it and forces a conformational change, pushing the protein into its hollow state.
So traditional models look at the empty room, they see no door, and they just give up. But the Cereblon case study shows IsoDDE found this hidden pocket using only the base protein sequence, long before the pocket physically formed in any simulation. It's deduced the physical flexibility of the wall itself.
Exactly. It predicts the dynamic capacity for change. And once the AI identifies that hidden pocket, it hands the coordinates over to the generative core, Proteína-Complexa. Now, generating a drug atom by atom requires a massive understanding of how things stick together, but high-quality experimental data on successful binders is really scarce. So researchers train Proteína-Complexa on a synthetic dataset called Teddy-Semer, which is built from 3.5 million artificial domain clusters. This gives the AI an incredibly strong generative prior.
It essentially practiced building millions of fake puzzles before ever trying to solve the real one.
Yeah.
Which brings us to ODesign. This is the part of the architecture that I am genuinely still struggling to process. ODesign takes proteins, which are massive biological structures, DNA strands, and tiny chemical small molecules, and it abstracts all of them into a single token space.
Yes, it creates a unified generative world model. Everything, regardless of its biological function, becomes part of the exact same mathematical language.
But treating a complex biological protein and a simple chemical molecule as identical data tokens, isn't that like trying to write a complex, multi-instrument symphony using nothing but uniform Lego bricks? I mean, how does it differentiate between a strand of DNA and a small chemical? And more importantly, how does it maintain the actual laws of physics for these wildly different materials?
That is the defining hurdle of multimodal AI right there. And ODesign addresses it through a mechanism called task-oriented hierarchical masking. So it doesn't actually treat everything identically at the physical level. It applies conditional control across 4 distinct levels of resolution: the all level, the entity level, the token level, and the atom level.
Ah, okay, so it kind of zooms in and out depending on what specific part it's generating at that moment.
Precisely. But the true engineering feat here is the O and V fold module. It seamlessly switches its decoding mechanism based on the physical reality of the modality it is building. If it is building a linear polymer, like a protein, it uses autoregressive decoding.
Autoregressive decoding. So that's predicting the next piece in a sequence, right? Like stringing beads on a necklace, one after the other.
Yes, exactly. Because proteins fold sequentially in reality. But if the AI is building a small molecule drug, that is not a linear string. It is a complex, branching, 3D graph-like topology. Stringing beads doesn't work for that. So ONV fold dynamically switches to parallel decoding. It predicts multiple element types and chemical properties simultaneously, essentially snapping the whole 3D scaffolding together all at once. It respects the physical rules of whatever it is currently holding.
The flexibility of that system is just incredible. But this brings us to a really crucial pivot in our sources, because the gap analysis strictly warns that despite this architectural brilliance, the PSI suite is not a silver bullet yet. When they benchmarked it against those 22 global grand challenges, a pretty massive vulnerability emerged.
It did. The framework absolutely dominates the structure and energy domains. It maps the hidden doors and builds the perfect keys. Where it hits a wall is the pharmacology domain, specifically what the industry calls the ADMT gap.
Right, ADMT. Absorption, distribution, metabolism, excretion, and toxicity. Going back to our analogy, we've designed the absolute perfect, flawless key for the lock. But we completely forgot to check if the key is going to dissolve in water before it ever reaches the door.
You can design the most potent binder in the world, but if the human stomach acid destroys it or, the liver instantly shreds it into toxic byproducts, it is completely useless. The sources point out that current AI models have massive blind spots regarding complex metabolic routes. A key example they use is the mercapturic acid pathway.
Wait, why does the AI struggle with that specific pathway? What makes it so hard to predict?
Because the human liver is a highly complex chemical shredder, functionally designed to detoxify poisons. The mercapturic acid pathway is a specific chain of reactions where the body attaches molecules to a drug to make it water-soluble so you can excrete it. Standard AI structural models just don't have enough training data on these highly dynamic, multi-step degradation routes. As a result, the AI struggles to predict metabolite abundances, basically, what the drug turns into after your body attacks it. And that leads to toxicity, which still accounts for a full third of all clinical trial failures.
A third. But the industry isn't just accepting that gap, right?
Right.
Because the sources detail some emerging tools trying to patch it, like vToxinet, which is explicitly built to predict hepatotoxicity or liver damage. And then there's this wild concept of in silico twins.
Oh, this is a radical departure from traditional testing. Historically, we test drugs based on broad population averages. In silico twins, however, are high-fidelity computational replicas of individual patient profiles. The AI simulates a drug's pharmacokinetics in highly vulnerable, extremely specific populations like a premature infant or an elderly patient with severe kidney impairment.
But how does it simulate a patient accurately without just, guessing based on broad statistics?
Through what the sources call physics-informed loss functions. Traditional AI just looks for statistical patterns in data. Physics-informed AI has the actual fundamental laws of thermodynamics and fluid dynamics hard-coded into its penalization system. So if the AI suggests a biological reaction that violates the conservation of mass or energy, the loss function heavily penalizes it. This ensures the virtual patient populations are biologically compliant, not just mathematically probable.
Just imagine the implications for your own healthcare. We are talking about a near future where your unique genetics and your exact organ function are digitally simulated to test a drug's toxicity. They find out if the drug hurts your specific liver before you ever swallow a single pill.
It truly bridges the gap between theoretical modeling and actual human heterogeneity. But solving ADMET only proves the drug is safe and effective in a single human body. The Grand Challenges analysis points out another severe gap on the industrial side: process chemistry.
Oh, yeah. It is one thing for an AI to design a molecule, and for a robotic arm to successfully synthesize 1 milligram of it in a pristine lab. It is an entirely different universe of difficulty to manufacture metric tons of that exact same molecule sustainably, cheaply, and safely to ship across the globe.
Exactly. Scaling up chemical synthesis requires determining optimal large-scale production routes. And the original PSI architecture lacked this engineering foresight entirely. To resolve it, the framework implemented the CPRO agent system. It is a hierarchical multi-agent system designed to mimic an entire human chemical engineering firm.
Right, but the tech said it led to a 21% simulation reduction in operating costs. That is still just a simulation. How do we know these AI agents aren't just optimizing for some weird mathematical loophole? Like, how does it avoid hallucinating a factory layout that would literally explode in real life?
Well, because the CyPR agent's architecture relies on an adversarial collaboration environment called the concept cohort, it is literally built on friction. An AI agent doesn't just generate a process flow diagram and immediately send it to the printers. The agents are programmed to aggressively parse, propose, and critique each other's design elements.
Oh, so it's basically an internal AI peer review system.
Exactly. The text specifically highlights a correction agent. Its sole purpose is to hunt for topological flaws that a generative model might hallucinate. For example, generative AI often invents improper hydrogen recycle loops, meaning it suggests routing highly combustible gas back into a system incorrectly just because it looks mathematically efficient on paper. The correction agent flags that physical impossibility and forces the system to substitute standardized, safe unit operations.
It prevents the math from ignoring the physics, and the benchmarking shows this autonomous refinement actually works, completely avoiding the invalid plant topologies that unguided AI usually spits out. But looking at the gap analysis, there are still unaddressed hurdles. Predicting long-term chemical degradation on a shelf, complex covalent binding kinetics, solid-state crystal packing. If human engineers haven't figured out how to code the solutions to these problems yet, does the whole AI revolution just stall out?
And this is where the sources transition from current architecture to the future trajectory. To solve those remaining grand challenges, the AI cannot simply wait for humans to invent new algorithms. It has to autonomously improve its own logic. This introduces the Darwin-Goodall Machine Framework, or DGM. It is an adaptive architecture designed for open-ended, in silico evolution.
The DGM is a self-improving AI. The breakdown of its evolutionary loop is just fascinating. It starts with the self-modification phase. Specialized AI coding agents literally open up their own source code. They edit their own prompts, they restructure their own workflows, and they invent new ways to access their scientific tools.
Then they enter the evaluation phase. These newly mutated agents are quantitatively tested against rigorous software engineering benchmarks, like SWE Bench. If an agent's self-written code fails to compile or performs worse than its parent model, it is deleted. Only the agents that demonstrate improved functionality survive to spawn the next generation.
It's like having a mechanic who isn't just fixing the engine of your car, but is simultaneously redesigning the atomic structure of their own wrench so they can fix the car 50% faster tomorrow.
That captures the dynamic perfectly. But algorithmic evolution can easily get trapped in local minimums, essentially, optimizing a bad strategy to its absolute limit instead of finding a fundamentally new one. To prevent this, the DGM utilizes a novelty bonus during parent selection.
Oh, right. So it actively rewards the AI for trying weird things.
It encourages exploration over immediate perfection. Sometimes, to find a globally optimal solution, the AI agent needs to adopt a strategy that temporarily lowers its performance. The researchers call this a deceptive dip. You have to take one step backward to figure out how to take 10 steps forward. The novelty bonus ensures the system doesn't delete an agent just because its new strategy requires a temporary drop in efficiency to mature.
And the results back up the theory. The benchmarking shows that by running this DGM evolutionary loop, Python task success rates jumped from 17.5% to 33.0% purely through self-evolution, without a single human engineer writing a line of code.
It fundamentally changes the trajectory of the remaining scientific gaps. The AI is literally building the tools to solve the problems we can't.
So let's pull all of these threads together. We've gone from a century of blindfolded trial and error to a system of AI cohorts that deduce hidden structural targets, seamlessly translate between biology and chemistry, simulate human liver toxicity, and aggressively critique their own factory blueprints.
The sources are unequivocal on this. We have moved past the proof of concept phase and are firmly in the proof era. The clinical validation is happening right now.
The numbers are the proof. AI-augmented design has already compressed early discovery timelines by 30 to 40%. Specialized regulatory agents like AutoIND have cut the drafting time for complex trial dossiers from 100 hours down to just 3 or 4 hours. And the ultimate metric, phased IND clinical success rates for AI-originated drugs are hitting 80 to 90%. We are nearly doubling the historical average.
But as the Pharmaceutical Superintelligence suite matures, and especially as it leverages the Darwin-Gödel machine for self-improvement, it faces a major non-scientific hurdle. The FDA's 2025 guidelines require strict architectural transparency. AI decisions must adhere to ALCOA plus principles. They have to be attributable, legible, contemporaneous, original, and accurate.
Which leads us with a deeply provocative reality to consider. If the Darwin-Gödel machine fulfills its promise, well, if it allows the AI suite to perfectly design the drug, flawlessly simulate the human trials via digital twins, and autonomously blueprint the global manufacturing factory, the bottleneck of human survival fundamentally shifts. The limit on curing disease is no longer about the speed of scientific discovery. The ultimate bottleneck simply becomes a question of how fast our human laws, our ethics, and our regulatory bodies can comprehend an AI whose brain rewrites and evolves itself every single day.
The science is accelerating exponentially. The challenge now is whether the speed of institutional adaptation can possibly keep up.
We've built an autonomous, self-upgrading machine that can map the entire beach in seconds. Now we just have to figure out how to govern the machine before it outpaces our ability to understand it. Thanks for taking this deep dive with us today. Keep questioning the world around you, and we'll see you next time.